Good afternoon. I want to thank the committee for having me speak today, and I hope to give you our perspective. And Dr. Cote and Dr. Neal, and Dr. Parasar, you've all heard this before, so I apologize for some of the repetition, but I want to hopefully put forth what my experience in drug development has said about what maybe are ways that we can practically and efficiently improve the rare disease review process. After about 11 years at BioMarin, a total 18 years of drug development, I started a foundation this last year to focus on these efforts and to try to do it, from what I've known, what could be improved. And we came up with the CureTheProcess campaign, which is really focused around 3 goals. Now, I want to point out that there are many things that you could try to improve, but we focused in on 3, which we thought were approachable or practical things which could be done that would make the process better. The 3 goals here: establish a new Office of Drug Evaluation. This is about specializing the review process and continuing to add additional expertise to the existing expertise, and there is significant existing expertise at FDA, but also letting that group work together as a leverage unit over time. We also believe improved accessibility, the accelerated approval pathway is important. I'll talk more about that. And the third area involves trying to improve study designs and statistical analysis methods to try to get the most power out of relatively difficult small studies, which are a great challenge and are often really need to be different from what we do in large market disorders. It's not working. There we go. So our campaign is formally endorsed now by 129 organizations. We had an 11 edition between the time the slides were submitted. And we continue to get support both from patients who are involved in a lot of rare disease projects, as well as from physician organizations like the Society for Immune Metabolic Disease and the American College of Medical Genetics. Oops. All right. So we know rare disease treatments are being developed and have been successful, and Dr. Goodman talked to them recently at the congressional hearing. Certainly a number have been developed for genetic disorders, so we don't deny that. The process can work for a number of diseases, particularly if they're large enough and if their clinical findings are amenable to treatment in the standard paradigm. Certainly drugs have gotten approved. But the problem is that there are a number of diseases where the paradigm doesn't work as well, either because they have difficult biology, like neurological disorders or bone disorders, or they're just so rare that there's not enough market incentive for the investment. For those situations, the process may not work as well, and those are the ones we're really trying to focus on. So we don't doubt that there are good drugs getting developed, but we do need to do more for some of these other disorders that are not getting treatment. Now, the one example I bring up commonly is slide disease, or MPS 7, and this has been successfully treated in animal models since 1993. And there's only about maybe a couple hundred worldwide with this disease, but clearly enzyme therapy in an MPS disease, I don't think anyone would deny that's likely to work in humans, since there are already 3 approved products for other MPS disorders. You know that's going to work, but who's going to develop it for that small of a market? But it could be done. If there's an accelerated approval pathway forward, it might be doable with a single trial approach. But that's not something that's been allowed recently. It depends on disease. There have been examples, maybe the one that was mentioned earlier, the RAGS deficiency, the urea cycle defect. But this is the kind of situation that shouldn't happen. If we've done all the science and we know how to treat it, we should somehow figure out the system to get them translated and make the system work for these kind of ultra-rare disorders. Now, my own experience helped build some of the issues I think that are important from hyaluronidase for MPS 1, in which the lack of experience in biochemical genetics, both at companies as well as at FDA, made it difficult to understand how to manage a disease like this. It was so rare. There wasn't enough regulatory science, in fact, for around the surrogate endpoints, or how do you decide if this is going to work or not? How do you interpret the data? What do you make of it? We couldn't solve the questions that were asked. The questions needed to be answered. And without those, we were not able to use surrogate endpoints, which I think dramatically changed what was going to happen in that program. We ended up with further issues relating to small clinical study designs and the right statistical methods, and being pushed into using a method that's commonly used for an endpoint, when in fact for a small study it should have probably been another method. And these are the integral tiny bits and pieces of regulation which are very difficult and which we need better guidance on, so that people will make the right decisions and not end up making mistakes in the process of doing drug development. Eventually, all this came up to an advisory committee vote, and the product was voted efficacious, and we did get approved. And then a 3-year delay occurred. But the more difficult problem is that the costs went up dramatically, probably 3 to 4-fold. And then, in the same time, some other products got knocked off our development path at BioMarin at that time. And those products still have no approved products at this point in time. Now it's been 10 years. And so there was a very significant impact when the costs go up. And this is not a blame of the FDA or anything. This is a situation how it occurs. I think what we need to do is figure out among all of us, scientists, FDA, and the public, what do we want for these kinds of endpoints? How do we want this development to occur? And we think that development of guidances and specialization at FDA would help that. Now, in terms of the Specialized Review Organization, the idea here is that there are people at FDA that are very experienced and knowledgeable in the rare disease area. And some of those should be allowed to be focused on rare disease type drugs. Now, that doesn't mean all rare disease. I mean, certain ones are not well covered. Some are very well covered in their divisions where they are. There are certain ones which I think are not as well covered, and I don't want to go into the scope issue right now. But by putting a group together, allowing them to work together as a team and stay together, the hope would be they gain additional expertise, leverage experience, and by bolstering that group with appropriate expertise, that we'd end up with a group that gets better and better, more knowledgeable about very difficult, complicated diseases. Now, we think one other part of this is that the workload on a group like this needs to be reduced because the applications are very complicated and there's a lot of unknowns. Whenever you're dealing with ambiguity, it takes more work, more effort, not less effort. And so that has to be accommodated. And I think we can't have people taking vacation in order to catch up with work as a reasonable solution to the workload issue. They have to figure that out. And the time they have to have is to be able to apply themselves to some academic links to NIH to also allow them to keep up with the latest of what's going on. That improved academic structure, we think, is an important thing, that they really need to be able to connect in with meetings and to have commissioner fellows that work with them. They're doing the hard nitty-gritty work of understanding what's going on in this latest technology area and how do I relate that to drug development. And the important thing then is it allows the FDA to be proactive in dealing with small biotechs that are struggling and trying to get somewhere. And I think the drug reviewers themselves must know the regulatory science issues. They can't rely on another group at FDA to tell them what they are. They really need to be able to understand them themselves because they have to sign on the dotted line. They're responsible. And they need to know them in order to be comfortable making what are very difficult decisions, because drug regulatory issues are complicated and always ambiguous and difficult at times. So we understand how challenging it can be, but they need enough time and sufficient staff to make this work. We'll go on to the next one. We'll skip this one. So let's talk about accelerated approval, the second goal. The accelerated approval regulations, I think, are a critical area for improvement. And the challenge is that the law says they need to be reasonably likely to predict clinical benefit. That's what they say. The question is, how do you interpret that? What does that really mean? And I think in the examples I have seen, the difficulty has been there's almost never enough clinical data to help you prove the case that it's reasonably likely to predict clinical benefit. And therefore, you're ended up, especially with some of these rare diseases, being unable to access this pathway. Now, the difficulty with surrogates is that, on one hand, the FDA is getting critiqued for approving things for type 2 diabetes, let's say, on a surrogate, and people are mixing up surrogate issues for large market disorders where there's a lot of other complexities with surrogates in rare disorders where there's a very unique, difficult situation for which it's not possible to do a survival study and where it's not possible to do the kind of work that you should be doing perhaps in type 2 diabetes. So because the surrogate endpoints are, and there's been a lot of attacks regarding using them in large markets, I think it's kind of caught up the whole rare disease area as well in that whole discussion, that debate. And I really think they should be separated, and that in a rare disease situation there should be a different take and look at how you look at accelerated approval. And it should be separate, and there shouldn't be any risk of a slippery slope between what you do in rare diseases with what you do for large market disorders where you need stronger evidence of proof and particularly safety. By improving the uncertainty exists today around a surrogate, you will turn on the investment of companies into this area. Because if they knew that there's these certain things that they need, this bit of data they should have and they should do the work on, if they have this set of data, that this is going to give them an acceptable surrogate, this allows them to start investment and begin doing drug development for some of these other rare disorders. Without that certainty, when they drop a what's a net present value calculation, how much money they have to invest over time, and they're using a clinical endpoint and the length of the studies and the time, they're going to come up with a number that's just too big, and it will never make any sense to do the investment, especially with the risk of failing. And that is what companies do. I was at a company, that was what we ended up doing. I'm working now with my intern to write a paper, which I'll show you, for a dozen drugs that clearly would work if translated to humans, that by looking at how if you apply a surrogate endpoint, you would dramatically reduce cost, so that, for example, with $1 billion invested in drug development, you would get 40 drugs approved versus 10 or 12 if you used a clinical endpoint. We have to understand how those factors, how the predictability affects investment, and that's how we can help accelerate pulling in money from elsewhere that's working on Me Too drugs that we don't need, and get them working on new drugs for things that are untreated. We think that can be done by creating good criteria. So one of the things I like to bring up, is that the accelerated approval pathway has been difficult to use, and that there's only been one genetic disease approved by this pathway. It's worked well for HIV and cancer. I know accelerated approval recently had some critiques. The truth is the pathway has really been extremely important in HIV and cancer. And there have been some genetic disorders, because it was brought up by Dr. Goodman recently. For example, the PKU drug, sapropterin for PKU, was approved on a biochemical endpoint, but it was given a full approval. But that's an unusual, that example, or that exception proves the example, the case, which is, in that situation, we had so much drug development data, or treatment data, from another treatment, that we could prove that the marker mattered. But we rarely have a situation where we have 20 other controlled studies to tap into to say, look, here's how it relates. That case just doesn't exist very often. And that's why it's difficult to use the accelerated approval pathway. Now, what we've been trying to propose, we need to go forward again. All right, so if you look at what's happened in HIV, there were 29 drugs approved in a period of 16 years with 6 different mechanisms of action. 6 different mechanisms, including ones no one even conceived of at the beginning. And that's because a bunch of little startups with a little bit of money were able to get a little data and blow out all the ideas that people had about how the treatment should occur and change the whole field. But that doesn't happen if they can't go to a VC and say, look, if I can do a 10-patient study and show I can reduce this marker, we'll have something that's movable. And that's how they get the money to make this happen. And that's how you take a disease from a death sentence to a managed disease. I also point out, too, there were 4 combination products approved. And how many disease areas have combination products approved in that same time frame? Very difficult to do without accelerated approval, without the marker endpoints, to do combinations and figure out how to do them well. This is why I think it would actually drive far better drug development than what we can with just clinically driven studies. We think there's some simple things we could do in qualification criteria for surrogates, and I'm suggesting a few here that would help give us the scientific confidence that this makes sense, that the disease is understood, the drug is understood, and why the marker you're using makes some sense. You shouldn't have to be able to have prior clinical data, because that just never happens. And that's where we end up getting hung up with MPS I. And I think it's just, we need to be able to figure out how to use the other parts of the science to make the judgment in a rare disease for which there might be only a few hundred people in the United States. The last area I want to talk about is analyzing clinical data in heterogeneous patients, and it's extremely difficult, maybe too arcane a topic, but we don't think that the current methods are often optimal, and the traditional approaches often are not very sensitive and are designed for larger patient studies where you get over the noise by simply having more patients. And that doesn't work well, and I think there are many times with drugs that are for rare diseases where you'd miss the treatment effect because the patients are so heterogeneous and it's almost impossible to get enough patients of a particular narrow slice and condition. And I think we need some better FDA guidance on what you could use for designs and analyses. And in order to help inform on this, I've assembled a scientific advisory committee. We're taking data from approved orphan drugs that we can get, and we're running analyses on that, which will look at different approaches, statistical analysis of a drug that we know is successful. And then we'll model it using the treated and placebo groups to help show whether the method was more sensitive and also retained specificity. That is, we don't want methods that generate false positive, drugs that don't work coming up positive. But we think by doing this analysis effort will help guide on what are the things that you can do in analysis of a small study or in design of it that will give you greater sensitivity and get the right answer that is showing efficacy when it really does exist. So these are our three goals: getting the right staff in place and helping give the staff that are here enough support and time to be able to do their jobs. And we think improved accessibility and predictability for getting into the approved accelerated approval pathway will be quite important. And finally, study design and statistical analyses. I think these three things would have an important impact in what we want. And we think what's going to happen if we get these things, more new drugs for rare disease, I think there's no question we can get companies to invest to make that happen. We think we'd get many more patients treated, particularly for diseases. For currently, we'll not ever get a disease developed. There are certain diseases that are just too difficult from a neurology basis or bone or whatever it is that they just won't go through because without these improvements. And finally, we think, in a time when the economy is poor, I think there's a lot of good high-paying jobs that are come out of these programs, and I think it's a factor understanding that this is an area where we can have a positive impact in doing good science, taking good science into great medicines. And this is why we hope that this commission, in doing its duties, will help find those tractable, approachable solutions. And we certainly hope that you get additional funding for drug review, and as well as help us find the way forward in getting drugs development for rare patients. Thank you.